Hello, everybody. So this has been the completion of the nucleic acid class. So we've got two types of nucleic acids. We've got deoxyribonucleic acid, which is DNA, and we've got a ribonucleic acid. So the DNA provides direction for its own replication. So the DNA directs synthesis of messenger RNA, mRNA, and through mRNA controls protein synthesis. As we all know, the DNA is a double helix and it's going to split into two to form mRNA. So the mRNA is the one that actually translates to become a protein. So protein synthesis usually happen in the ribosomes. Ribosomes is part of the cell, which is in the cytoplasm. So this is why the DNA will come out of the nucleus. So then it becomes an mRNA. So, and it moves out of the nucleus to the cytoplasm region into the into the ribosome, where it's going to produce the proteins, which is called polypeptides. So the nucleic acid are polymers called polynucleotides, because nucleic acid is actually a combination of nucleotides. So each polynucleotide is made up of monomers of nucleotides. So if you have a nucleic acid is made up of group of nucleotides. So this is something unique we have to understand. Each polynucleotide is made up of monomer of nucleotides. So the meaning is polynucleotide is synonymous to nucleic acid. Each nucleotide consists of nitrogenous base, pentose sugar, and a phosphate group. Pentose sugar is a kind of sugar which is got 5 carbon chain. So this is why it's called pentose. Nitrogenous base, they are the UCG and the T, while the phosphate group is the number of phosphate we usually know. So the portion of a nucleotide without a phosphate group is called nucleoside. So if you have a phosphate group, if you have a nucleotide which is lacking the phosphate group, is called nucleoside. So now polynucleotide or nucleic acid, they are they are in this structure. We have a 3N and we have a 5N. So the 3N is here. This is a pentose sugar. If you count from this side, you count 1, 2, 3, 4, 5. So connecting with another phosphate group. So this is a nitrogenous base, and this is a pentose sugar. This is a phosphate group. So this is what makes up the nucleotide. This combination is continuous. From you have a 5N, you have a 3N. So the nitrogenous base is divided into two. We have the pyrimidines, and we have the purines. The pyrimidines, they are the ones which are the cytosine, thymine in DNA and uracil in the RNA. And the purines are adenine and guanine. So we've got the sugar molecules. The oxyribonucleic acid is the OH groups on the pentose sugar lose one of the oxygen. So this is why we call it deoxy. It lost oxygen. And the ribose in the RNA has its oxygen molecule. So this is the same picture. So this is a same picture for the nitrogenous bases. Now, nucleoside is equal to the nitrogenous base plus sugar without a phosphate. I want you to remember this. There are two families of nitrogenous bases. We have the pyrimidines. The pyrimidines consist of the cytosine, thymine, and the uracil. So what is unique about the pyrimidines is they have a single 6-membered ring. So they're not doubled ring. With the purines, which is the adenine and the guanine, they have a 6-membered double ring fused to a 5-membered ring. So I want you to always remember this. So the purines have a double ring, while the pyrimidines have a single ring. In the DNA, the sugar is deoxyribose, in RNA is ribose. So the nucleotide is equals to the nucleoside plus the phosphate group. Remember, the nucleoside is a nitrogenous base with sugar, but the phosphate is not there for the nucleoside. So now with the phosphate group, you have a nucleotide. So nucleotide polymers are linked together to build a polynucleotide. So adjacent nucleotides are joined by covalent bonds that form between the OH group on the 3 carbon of one nucleotide and the phosphate group of a 5 carbon on the next. So this concept is very, very important. So adjacent nucleotides are joined together by covalent bond, which is formed between the OH group of the 3 end carbon of one of the nucleotide and a phosphate on the 5 carbon on the next. So this links create a backbone of sugar phosphate unit with nitrogenous bases as appendages. So the meaning is the main backbone is sugar and phosphate unit. So the nitrogenous base is just appendages, which is not a very important part of it. So the sequence of bases along the DNA or mRNA polymer is unique for each gene. So we will talk about that later. So a DNA molecule has 2 polynucleotides, spiral link around an imaginary axis, forming a double helix. So a DNA molecule has 2 polynucleotides. So these 2 polynucleotides, which we call double helix. So the spiral around an imaginary axis, so forming a double helix. So in the DNA double helix, the 2 backbones run in opposite direction, 5 to 3. So the directions from each other and arrangement referred to antiparallel. So the meaning is, if you have a DNA molecule, like you've got a DNA molecule which is ACGT. So A is going to run this way and ACGT will run other direction. So one runs this way, the other runs in the other direction. So this is the meaning of antiparallel. So one DNA molecule includes many genes. So the nitrogenous base, the DNA, pair up and form a hydrogen bond. So the adenine is always with the thymine. So we usually have a T relationship. So every time you have adenine, you have it paired with thymine. While the guanine will always pair with a cytosine. So this is what they mean by the pair up. So it's a pair. For example, let's say you have a CGTCA. So if it's going to pair up, it's going to be, this is going to pair up with a T, and it's going to pair up with a G, and this is pairing up with A, and this with a C, and this with a G, and this with a T. So this is exactly what they mean pairing up. So now this is a 5' and a 3'. So this is called the sugar phosphate backbone. So this is the major backbone that make up the DNA. So this is a sugar phosphate backbone. So the inside is made up of the nitrogenous bases. So base pair joined by the hydrogen bond. So what joins the bases together is a hydrogen bond, and you can see A with a T, C with a G. So and you would notice if you go back, you will notice the pyrimidines. So C, T, and U are the pyrimidines, while the purines are A and G. So this is why one single ring will bond with a double ring. So that's why you have a CG, and one double ring bonds with a single single ring. So that's why you have AT. So this is something you need to understand very well. So, and as we mentioned to you before, so the old strand nucleotide about to be added to the new strand. So what would happen is when there is a kind of genetic where you want to grow new genes. So what usually happen is you have a 3N and a 5N. So these are all strands. So the old strands would unwind. So when they will unwind, they will form two new ones. So this is one, and this are 1, 2 from the same. So from the same DNA. So when you have a DNA for G, T, A, C, T, G. So this is gonna have a C. This will have but T. This will have a this will have a T. This will have a G. This will have an A, and this will have a C. So when they want to be genetic growth, or they want to be the gene wants to division. So you're gonna have a C and a T and an A and a C and a T and a G. So then you're gonna have a C and A and T, G, A, C. So this will pair with G, this will pair with T, and this is gonna pair accordingly to C. So this is exactly what we call when the cell divides. So during cell division, one DNA molecule would grow to become two DNAs. So this is exactly what happens during during the cell division. So DNA and proteins are step measures of evolution. So the linear sequences of nucleotides and DNA molecules are passed from parents to offspring. So the sequences we have in the DNA molecules, nucleotide, what we saw before, they always pass from the parent to the offspring. The meaning is they conserved, they managed properly and sent from parents to the offsprings. Two closely related species are more similar in DNA than are more distantly related species. The meaning is when each species are more related to each other, they have a more similar genetic makeup than the ones which are far away from each other. So molecular biology can be used to assess evolutionary kinship, which we're going to look into very well in this class, in this AP class. So higher levels of organization result in emergence of new properties. So the meaning is when organisms move from low level to higher level, then there will be emergence of new properties. There will be new things which is going to be coming out different from the ones you had in the beginning. So organization is a key to the chemistry of life. So now this is a summary of what we've done. So we started with the biological molecules. We started with carbohydrates. So carbohydrates is referred to as the fuel and building materials. So and carbohydrate is COH, is made up of carbon, oxygen, and hydrogen. So, and this is the monosaccharide molecule. So it's got 1, 2, 3, 1, 2, 3, 4, 5. So if you want to number it, 1, 2, 3, 4, 5. So this is the sixth one. So the sixth one is actually where it starts from. So when you're counting it, you count from there. So, but sometimes you can have the line switch it. Instead of coming this way, it goes straight. So you count it straight once you know it's part of the carbon. So what all this we're counting is a carbon background, nothing. So monosaccharide, glucose, or fructose. So the monosaccharides, the one which are simple ones, are the glucose in normal human being, in normal breaking down of food, and fructose in fruits, sugar got in fruits. So cellulose disaccharides are the lactose and sucrose, and polysaccharides are cellulose, starch, glycogen, and chitin. So the function of carbohydrate is a few carbon source that can be converted to other molecules or combined into polymers. So they strengthen plant cell wall. This is a cellulose. The starch helps to store glucose for energy. The glycogen store glucose also for energy in humans and animals, but this is in plants. So the chitin helps to strengthen the exoskeleton and the fungal cell wall. So the second one is a lipid. So lipid is a diverse group of hydrophobic molecules and are not macromolecules. So the glycerol is this, you got three fatty acids. So this is the reason why you call it triacylglycerol. So which is fatty oil. So you got glycerol, you got three fatty acid. So this is exactly the composition of the word of lipids. So if someone says, what are the composition of lipids? Say glycerol and what? And three fatty acid. So this is important source of energy. So, and add with phosphates and two tails. So this is what the composition of the cell membrane. So we call that phospholipids, phosphate group and two fatty acid. So these are the lipid bilayer of the membrane, which we talked about in the class. So we also have a steroid backbone, which is this. Steroid is 4 fused rings with attached chemical groups. So 1, 2, 3, 4. So it's rings being fused together, so and attached together by chemical groups. So the components is a component of cell membrane, the cholesterol, and it's a signal that travel through the body, the hormones. So the proteins, which is the third molecular, large molecular biological molecules, which are important in the body. So proteins have many structures resulting in a wide range of functions. So if someone asks also the component of protein, say amino acid, the R group, and the what? And the carboxylic group. And the carboxylic. So this is it. This is 1, this is 2, this is 3. So we have enzymatic, we have enzyme protein, they catalyze chemical reactions, structural, they give support, storage, they store amino acid, transport, they move things, hormones, they have to coordinate organismal responses, receptor proteins, motor, and defensive, which help to protect you against disease. And the fourth one is a nucleic acid. A nucleic acid is composed of phosphate group. I want you to understand this. Phosphate group, a nitrogenous base, and a what? And a sugar molecule. So DNA is sugar. The sugar is deoxyribose, while the RNA is ribose. So nitrogenous base is CGAT in DNA, while usually double-stranded, and the RNA is CGA. The T is replaced with uracil. So, and usually what? Single-stranded. So I want you to understand this concept very well. Let me remove this. So the T is being replaced by U in the, if someone says, what is the difference between the DNA and RNA? So the DNA is double-stranded. This is deoxyribose, it lose one of the OH molecules is lost, is oxygen. So that's why it's deoxyribose. So usually double-stranded, but the RNA is got the ribose sugar completely. There's no oxygen lost in it. So the nitrogenous base, the U is replacing the T, and also is usually single-stranded. So carries protein coding. It carries the protein coding, which, I mean, what do you mean protein coding? C, U, A, G, C, G, T, C. These are the three molecules, or I say the three nitrogenous bases that usually form a protein molecule during the process of translation. So we would have more time to talk about that, inshallah. So this is a repetition of the same thing. So this is why the slide is so voluminous. So now this is glycosidic linkage being broken. What does this mean? So when a glycosidic linkage between two monomers of carbohydrates are being broken, what would happen? So they form monomers. So this is a linear form of your carbohydrate. This is 1, 2, 3, 4, 5, 6. So C6. So when you count C6, H12. So you want to have your H12. So you have OH on this side. 1, 2, 3, 4. So this has been replaced with double bond. So the OH is gone. H is gone here. H is gone here because of double bond. and this. So you will count H 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 12. Or 6, 1, 2, 3, 4, 5, 6. So this is A. So this is the distribution of OH. So, and you have HH in all the side, you have OH, yeah. So this has been swapped. So the third carbon is swapping the H and the OH. So if you want to form the same thing in the ring light structure, what you just need to do, just pick up one side, maybe the 6, or pick up one, fold it, bend it like this. Then you have, when you bend it, you're gonna have your C, then you have your you have your OH, and you have your H, and you have your you also have your H. So instead of having HH and all this stuff, you can have your CH2 and OH. So this is why you have CH2 and OH here. So this is the second one. So the second one is called the double oxygen, we should say. So then the third one is got H and OH, and the fourth one is got OH and OH, and the fifth and the sixth. So this is a simple thing. So, and also this is a ring light structure. This is the same way it's been formed. So I want you to always remember that the same thing you have here is what you have here. There's nothing different in it. So you can decide to pull these all of this together in CHOH. So CH O CH, you can say CH2. This is 1 and this is 2. And remaining this OH. So this is how you have your CH2OH. It's nothing special in it. All right. So this is exactly what I want to explain to you. So now the second one is the you've got the carboxyl group here, and you've got the amino group. So, and you've got the sulfur okay, there's a peptide bond joining one amino acid with another one. So, and this carboxyl group is, like I mentioned to you, is always one of the content of amino acid. So you have amino group, which is this, and you have the R group, which is this. You have the carboxyl group. It's always like this. So the bond connecting one amino acid and the other one is called the peptide bond. So this is exactly what it's telling you here. So when you remove the water molecules from two monomers, so they become a polymer. So this is exactly what's happening here. So now let's move on to the next one. So this is the 5N and the 3N of the DNA. So now this is exactly what I was talking about all the time. So here is a 5 prime. You've got a TAAGCCT, and it's a 3 prime. So the other strand is going to be opposite. It's going to be the 5 strand. We'll start from here. So T is matching with the A, C with G, C with G, G with C, A with. So in this case, this is not matching. A is supposed to match with the one with a T. So A, T, and TA. So now you've got a mismatch here. So this is what a mismatch is. A mismatch usually happens when there is no matching between the genetic makeup. So when you're supposed to have a T, you've got a C. So it's been replaced by a C. So it's a mismatch here. So TA, C, G, C, G, G, C, A, T, A, T, A, T. Now this is amazing. There's no mismatch here. All of it is fine. So now this is a summary of what I talked about before. The sugar is a monosaccharide, a monomer. The polymer is polysaccharide, is being linked together by glycosidic linkages. The lipid is the monomers of fatty acid. The polymers are triacid glycerol, and they be linked together by ester linkages. Remember these links, please. Proteins are monomers of proteins are amino acids. So the amino acids are being linked together by polypeptides. So the type of linkage is called peptide bond. So polypeptides are polymers of amino acids. So a nucleotide is a monomer of polynucleotides. Polynucleotide is the same as nucleic acid, and what is linking them together is phosphodiester linkages. So phosphodiester linkages is linking them together. So they say we need to original strand with 3 prime, and this is a 5 prime. So it's just trying to structuralize the DNA. So You will see in the DNA, this is the phosphate group. These are the nitrogenous bases, the G. And what's the third one? The nitrogenous bases are the ribose sugar. So the ribose sugar is 1, 2, 3, 4, 5. This is the ribose sugar, and this is the nitrogenous basis. So this is exactly what makes up. As you can see, this is phosphate group. This is 1, this is 2, 3, 4. This in this case is a double. So the guanine is a double. So this is why it's double ring, but the other ones are single ring. So this is exactly what we need. So if someone asks you, you should be able to listen, describe the 4 major classes of molecules, which are carbohydrates, proteins, amino fatty acids, and nucleic acid, nucleotides. So describe the formation of glycosidic linkage and distinguish between monosaccharides, disaccharides, and polysaccharides. It's simple. Distinguish between saturated and unsaturated fat, between the cis and the trans. Describe the 4 levels of protein structure. I believe all of this you should be able to answer them without problems. Distinguish between the following pairs: pyrimidine and purine, nucleotide and nucleoside, ribose and deoxyribose, the 5 and 3 prime, and end of the nucleotide. Okay, so this is the end of what we have for the remaining section of the molecular chemistry of life. So we're going to move on to the cell and the structures in the next class, inshallah. Barakallahu fikum.